having me. By being here, or by being on tape, you just ensured yourself that you're going to pass. Okay? You're going to pass the biochemistry portion of the boards. So just get that locked in your brain right now. So what I'm going to do today is demystify the mountains of notes that you guys have for those 2 biochemistry classes. Okay? So that's my goal. And I'm going to pare it down to what I think is the most important and what you're going to see on the boards. And I have a very good idea of what you're going to see on the boards because I reviewed boards tests. So this is the stuff. Now, that's not to say there's going to be a lot of details, but we'll just hone in on what's important. But for you guys, what you need to do is just realize that biochemistry is really just 4 topics: proteins, carbohydrates, fats, and nucleic acids. And that's it. That's all we're going to talk about. Throw in vitamins, which can be tied into proteins a bit, and that's a 5th. Okay? And for each one of these 4, what we're going to talk about is the structure and the function. And that's really Biochem 1. That's what we did in Biochem 1, structure and function. And then in Biochem 2, we're going to talk about their digestion and their metabolism. So all I'm going to do is just go through the very basics, the structure, the function, the digestion, and the metabolism of proteins. Then we'll do it of carbohydrates, fats, and nucleic acids. And the way I think I'll probably break it up is we'll do structure, function for all 4 first, and then we'll go to digestion and metabolism, which is Biochem 2. All right? So let's talk about the proteins first. Now, what are the structure of proteins? What are they composed of? Amino acids, all right? So, good news is, well, bad news is, these are all the amino acids, all 20 of them. The good news is you don't have to memorize their structure. Okay, what you do have to know are some of the facts and how we categorize these. So, to do that, let me just start giving you the general structure of any amino acid, and then we'll start getting into what you need to know about them.
Dr. Spear,
Yeah.
Can you align that so it's on the right edge of the screen? Just turn the overhead projector a little bit, or move. Yeah, there we go. As far as you can would be best. Okay, that's good. Thank you. Yep, perfect.
Okay, great. Now, so I'm just going to give you a little intro and then we'll get right into the details. Every single amino acid has an amino group and an acid group. That's why it's an amino acid. Amino and an acid. Every single group has what's called the alpha carbon, which the amino and the acid group are attached to. Every single amino acid also has a hydrogen attached to that. Okay, so all the amino acids have this alpha carbon, an amino group, an acid group, it's a carboxylic acid, and a hydrogen. The only difference between the amino acids is the 4th group, which is attached, and that's called the R group. Okay, so the only difference between all 20 amino acids is what they have here. It's the R group. Okay, so when we talk about putting amino acids into groups, it's going to be based on what this R group is here. Alanine's nonpolar, it just has this methyl group here. Now, all amino acids, when they're alone in solution, can exist in what's called either the L or the D form. And what that simply means is that there's rotation around this alpha carbon between these 2 groups, the amino and the hydrogen. They can exist on either side. Depending upon what side they exist on, what that means is they can rotate polarized light. So there's L or D. So with all of that said, what's the most important thing to remember? The most important thing to remember is that glycine does not exist as L or D. All the other 19 amino acids exist as L or D. Glycine is just glycine. So one of the questions on the boards you'll see is which name is wrong. It'll say D-alanine, L-alanine, D-alpha, phenylalanine, and then it'll say D-glycine, D-glycine. Okay. The amino acids can exist in either form, except for glycine. All right. Now, what I'm going to do is let's put the amino acids back up there, and let's talk about their properties. Okay, let's start up at the top. Valine, leucine, and isoleucine. Those are the branched chain amino acids. You want to lock that into your brain. Valine, leucine, and isoleucine. And if you look up here, you'll see that their R group, they're branched hydrocarbons. That's why they're the branched chain. One of the important things about the branched chain amino acids is that they're metabolized primarily in the muscle. That's why bodybuilders take them. The muscle metabolizes the branched chain amino acids. Look at the R groups of phenylalanine, tyrosine, and tryptophan. Right here. They all have rings. Those are the 3 aromatic amino acids. So these are just facts you need to lock in. Okay? Phenylalanine, tyrosine, and tryptophan are the aromatic amino acids. Classic question. Phenylalanine and tyrosine are interrelated. Phenylalanine can be converted to tyrosine. That's very important. It's important that you know the enzyme which does that. And the enzyme is called phenylalanine. Okay, so that's the substrate, hydroxylase, because it puts a hydroxyl group on it. Phenylalanine hydroxylase. The deficiency of that enzyme results in the classic metabolic disease known as phenylketonuria, or PKU. Phenylketonuria. Does this stuff ring a bell? Somewhere way back, yeah. Okay. But this is the stuff you need to take with you.
Question?
Yeah.
What is that thing that transfers phenylalanine?
It converts. It's the thing. Is the enzyme.
Yeah.
Okay. Phenylalanine. Okay, so that's the substrate. Hydroxylase. That's what it does. It adds a hydroxyl. The disease state when you do not have this enzyme is phenyl ketone urea. You guys have heard of this, right? In the other classes. PKU. It is the classic metabolic disease. The classic. So just lock that in. Lock that in. Okay. Another common question. What are the sulfur-containing amino acids? Cysteine and methionine. Cysteine and methionine both contain sulfur. And then they throw in a third one called cysteine. Cysteine, C-Y-S-T-I-N-E. And cysteine is formed between 2 cysteine residues when they form a disulfide bridge. So if you have a protein which is folded around like this, and there's a cysteine over here and a cysteine over here, what they do, if you look here, they both have H's. They lose those H's and they bind with oxygen to form water. And the cysteines form a covalent bond between each other, and that is called a disulfide bridge. And the residue, when that forms, is called cysteine. And so it's considered that there are 3 sulfur-containing amino acids: cysteine, cysteine, and methionine. Can you see that? Another word about methionine, and this is cell physiology, which we'll be incorporating a bit. Methionine is the first amino acid in proteins. It's what's called the start codon. Encodes for methionine. So methionine is the beginning of translation. Translation begins with the amino acid methionine. Now, what I want to talk about next, so that's really the classifications. And you guys that have my handout, you'll read there's a couple more diseases in there and just a few more things. Just focus in on those two. But what I just told you is the main thing you need to know about the amino acids. You also need to know what, so now we're going to talk a little bit about metabolism. What do the amino acids, what are they precursors for? Because other compounds can be made from the amino acids. So I've already told you that phenylalanine can be converted to tyrosine. So that's one amino acid to another. What's tyrosine the precursor for? Several things, right. L-DOPA. So in order, L-DOPA, DOPA, dopamine. Yeah, then we get into the catecholamines. Norepinephrine, epinephrine. Okay, the latter two are considered the catecholamines. So tyrosine is considered the precursor to the catecholamines, as well as L-DOPA. Okay, neurotransmitter. So you have tyrosine as a precursor for a neurotransmitter and the catecholamines.
Dopamine is not a catecholamine?
I think I wrote that on your sheet. I'm still unclear about that. Don't
Go with norepinephrine and epinephrine.
Yeah, and don't spend a lot of time looking that up. Now, there's one more thing that tyrosine's a precursor for, and that's melanin, your pigment, because melanin is derived from L-DOPA. So originally, bless you. So it's tyrosine, L-DOPA, dopamine, norepinephrine, norepi, and then epinephrine. And then melanin comes off L-DOPA. An enzyme which you may see, which is important here, this first one, and it's called tyrosinase. And tyrosinase is the enzyme which converts tyrosine to L-DOPA. Now don't lose sight of the fact that because phenylalanine is a precursor to tyrosine, phenylalanine is also a precursor to all of these, but it's usually considered that tyrosine is the main precursor. But the advantage that gives us is if for whatever reason we're not taking in any tyrosine in our body, if we're taking in phenylalanine, we're okay. But people who do not have phenylalanine hydroxylase can't do this, and so they must take in tyrosine. They must take in tyrosine. Any questions on that? So you need to know some of the main precursors of these amino acids. Tryptophan. Tryptophan is a precursor to two main things. One is another neurotransmitter, serotonin. Serotonin. And then it's also a precursor to the coenzyme, or not to the, it's a precursor to niacin. Let's just put it that way. So tryptophan goes to serotonin. It also goes to niacin. And what's niacin the precursor of? Okay, so niacin you should think of B2. No, doesn't go that way. Niacin, which coenzymes are made from niacin? All right, good. So we know where we need to go. Tryptophan goes to serotonin. That's one. And then it goes to niacin. And if you, so we're just going to jump ahead a little bit, but this is okay. Niacin is B2. Excuse me, I'm sorry.
That's okay.
Is that what you said
It's
B3
early.
before?
Yeah.
I'm sorry about that.
It's early.
You're right.
That's okay.
Thank you. Good catch.
You talk.
Niacin's the precursor for NAD plus and NADP plus. Okay, these are the two coenzymes. Coenzymes. This is the vitamin. Very good. And remember, when I write NAD plus, that's just the oxidized form. The reduced form is NADH. The reduced form here is NADCH. Oxidized without electrons, reduced with electrons, and the electrons come attached to the hydrogen atoms. Oxidized, reduced. So that tells you right there that NAD and NADP plus are involved in oxidation reduction reactions. And they are derived from niacin. And if we don't have niacin in our diet, but we have ample levels of tryptophan, we're okay. All right. Cool. Histidine. Very simple. Histamine. The histamines are derived from histidine. So in the boards, you're going to be asked to really funnel down this huge, broad, vast array of information down to facts like this. Okay, and then hopefully for those of you who are taking the board review, I know Dr. D'Onofrio is going to take all these facts and actually bring them together in some type of cohesive, manageable way. Okay, but right now, from my standpoint, just what I want you guys to do is not spend hours and hours and hours focusing on biochemistry when you're focusing on the wrong stuff. Commit this stuff to memorization, you'll do fine. Okay, so that's it for the amino acids. You're well on your way if you know that material. Well on your way. So what happens to the amino acids? How do we go from amino acids to proteins? Peptide bonds. Very good. Amino acids get linked together via peptide bonds. Okay, so I'll just draw one example of that. NH3C, this is the alpha carbon. You have the R group, which is variable, the hydrogen, and then the carboxyl group right here. What I'm going to do is draw the peptide bond. This is amino acid 1. And here comes amino acid 2 with its amino group. The amino group reacts with the carboxyl group to form the peptide bond. Okay, this is the peptide bond. It's this guy right here, but it's involved between the carboxyl group of one amino acid and the amino group of a second amino acid. The enzyme which catalyzes this is peptidyl transferase. Peptidyl transferase. And all a protein is, or a polypeptide, is just amino acids getting linked together one at a time by the process of translation. Process of translation. Just linking the amino acids together via the peptide bond. Now, again, I remember we went over this class in several quarters for you guys, but this is an amide bond, but when you're talking about proteins, it's a peptide bond. So a peptide bond is a subset of the amide bond. This is an amide bond because these are the two groups which form an amide bond, But when you just think about what a protein is called, it's called a polypeptide. So it's a peptide bond. And this is the enzyme, peptidyl transferase. Any questions on that? Right into proteins. There are 4 levels of protein structure. Okay, primary, secondary, tertiary, quaternary. Doesn't get more complicated than that. Very straightforward. Primary is just the linear sequence of the amino acids. That's it. The secondary structure, there's just 2 types. This classic question, okay? Alpha helices are a type of what? And you should say secondary structure of protein. Okay? The 2 types are alpha helices, or an alpha helix, the helices, and then beta pleated sheets. Even if you have no idea what the hell these are, if you remember that these are types of secondary structure, you get the question right. Half the other people in the country miss it. Okay? If you want to look this up to get it clear in your head, to lock it in, go ahead. But this is what you need to know. Tertiary structure is simply, there's 2 things you need to know about it. The first is it's a single polypeptide. You're only talking about one polypeptide, single polypeptide. Which folds. Okay, so it's just the folding of a single polypeptide. And these are what are called globular proteins because they fold into globs. The classic example, which we spend a fair amount of time on in class, is myoglobin. So let's do a little cell physiology. What's the function of myoglobin? Holds oxygen in the muscle. And where is it located in the muscle? Which organelle in the muscle? Mitochondria. Very good. And why does it make sense to have an oxygen storage in the mitochondria of the muscle? Right, because that's where the electron transport chain is. And the electron transport chain requires oxygen, and that's where you make all your ATP. Exactly. So if you can just think it through like that, you're great. Okay? Quaternary structure is nothing more than multiple polypeptides coming together. Okay, so it's the association of more than one polypeptide. Greater than one polypeptide. Classic example is hemoglobin. Hemoglobin is a tetramer, so it has 4 polypeptides. 4 polypeptides. It's alpha 2, beta 2. Alpha 2, beta 2. And what you see is, this is the classic theme in biochemistry. Structure determines function. And as you increase in structural complexity, the body increases in functional complexity. So myoglobin just sits in the mitochondria and holds on to one oxygen. Hemoglobin travels through your bloodstream to your lungs to drop off CO2, pick up oxygen, goes to the capillaries of the tissue, hands off oxygen, picks up the CO2. It's much more complicated. And that's because it has a much more complex structure. And that's really it. When you think of hemoglobin as something I'm not going to talk about much today, you should review what allosteric means. Allosteric effectors and allosteric. Usually allosteric enzymes are what come in play. Hemoglobin is not an enzyme, but it does have allosteric effectors. Also, cell physiology-wise, good thing to remember, and you should, I know you guys have got this in some of your other classes, the Bohr effect. That's a good one to know. Want me to go over that real quick? Okay. All the Bohr effect is, this is what allows hemoglobin to drop off oxygen into active muscle. That's the bottom line. How does it come about? When you have actively working muscles, you have an increased production of CO2 from the Krebs cycle, and you have a decrease in the pH from both lactic acid and the bicarbonate buffer system. An increase in CO2 and decrease in the pH facilitate the release of oxygen from hemoglobin. So when the hemoglobin encounters a capillary where the muscles are really working hard, there's going to be a lot of CO2, the pH will be very low, and the hemoglobin is just going to give up all of its oxygen right there. Okay, that is a type of allosteric effect, the CO2 binding, and that is called the Bohr effect. Okay, that's it for protein structure. So we've already gone through all the amino acids and the types of protein structure. When we talk about the function of proteins, which class of molecules are we going to talk about? When we talk the function of proteins, what's that? Enzymes, exactly. So this is a big class. This is a big part of the, not a big part, but a part of on the board. Most of the enzymes you'll have to know are going to be metabolic enzymes, which we'll get to in the second part. But what I want to talk about now is the characteristics of all enzymes. Characteristics of all the enzymes. While I'm looking for this overhead, are there any questions thus far? You guys just have this stuff cold? Too early on Saturday morning? All of the above? All right. Sure.
Are you going to be going over titration of PK?
No, I'm not.
Is that important? Do you suggest we do?
Okay, what I suggest is you understand what pKa means. And I'm happy to do that at the end if people just want a quick refresher.
Okay.
All right?
Never heard you give a lecture without PKA.
I know,
Just
really.
never have.
You feel deprived, huh? All right, yeah, we'll talk about pKa right at the end. I have not seen much of it on the board. I've seen a little. Okay, what's the main thing which enzymes do? They catalyze reactions, and when we use the word catalyze, what does that mean? They increase the rate. They increase. Kevin, you could probably teach this thing. You've done so much tutoring. Enzymes increase the rate of a reaction. And so what you need to know, number one, is that's what they do. And so the next logical question is, how do they do it? And that's what this curve is all about. Enzymes increase the rate by decreasing what's called the activation energy. Activation. They decrease the activation. Let me just briefly go over that. And there's going to be a couple terms that are going to come up here which you really should know. All right, I'll stand over here. What this shows you is the free energy. Remember that? G for Gibbs free energy. And this is the progress of the reaction. What you have is some reactant, so it's A. Okay, you don't care what it is. And A has some energy. For the reaction of A going down to the product, which will be B, so we're just looking at A going to B, you'll see that the product is lower in energy. It's lower in energy than A. And so what you do, so I'm introducing a couple terms here. So to look at the delta G, which is just the change in energy, you're always just looking at the substrate versus the product. You don't care about the pathway. And when the product is lower in energy, that means it's a negative delta G. That means the reaction is favorable, spontaneous, and exothermic. Negative delta G, favorable reaction, spontaneous, exothermic. Okay, this is just a fancy way of saying the product has a lower energy than the substrate. Okay, so So you started off on the 5th floor and you've walked down to the 1st floor. Okay, if you jump off the 1st floor, you're not going anywhere. If you jump off the 5th floor, you've got a lot of energy. Okay, so you're just lowering the energy. However, for the reactant to get to the product, it has to go over this huge hill. And the huge hill is the activation energy. And in the absence of an enzyme, all the reactants have to climb that hill. In the presence of an enzyme, the enzyme just burrows a tunnel right through, and it's much easier for the reactant to go to the product. So with a catalyzed reaction, the activation energy is lowered, and the reaction happens at a faster rate. Okay? If you lock that in, every enzyme works this way. That's the energetics. Let me just do the flip side on the delta G. Okay? If the product is higher in energy, that's when you have a positive delta G. That's when you're walking up the stairs from the ground level to the 5th floor. That's a positive delta G. That's an unfavorable reaction, non-spontaneous, and it requires an energy input. So it's endothermic. Just rephrase that, restate that. Positive delta G, non-favorable, unfavorable, non-spontaneous, endothermic, and it requires an energy input. That means when you're thinking of biochemistry, ATP is coming into play. ATP will be cleaved to drive that reaction forward. Are you just reliving the excitement? Okay. That's it for enzymes. All enzymes work this way. They lower the activation energy. So that's it for this part of it. Okay, what are the two parameters of enzymes that you should look at for every enzyme? It's not allosteric. It is... There's two things that you guys hated. One was pretty easy, but the second was Km and Vmax. Well, there's so much to choose from. Km and Vmax. Okay. Km and Vmax. So for every enzyme, whenever one is studying an enzyme, you need to know what Km and Vmax are. So for every enzyme, it's going to have its own characteristic Km and its own characteristic Vmax. Vmax is the easiest thing. Okay. Here's how you look at enzyme catalyzed reactions. This is substrate concentration. So you just start adding a lot of substrate, and then you look at the velocity or the rate of the reaction. It's substrate versus velocity. So usually as you add more substrate, the enzyme is going to be able to bind more, and it'll go faster. And that's exactly what you see. Look up at this first line. You see this hyperbolic curve? And when you get to the point where you've added so much substrate that it levels out, even if you add more substrate, there's no change in the velocity, Vmax. Okay, so it's the fastest the enzyme can go. What it means is, let's say you have 100 molecules of enzyme, and when you're down here, you've only added 10 molecules of substrate, 20, 30, 40, 50, 60. When you get up to the Vmax, you've added 100 and above. And what that means is you've just saturated the enzyme. There's no more free enzymes, and it's going as fast as it possibly can, and that's where you reach the Vmax. Okay, so that's just one property of all enzymes. You look at the Vmax. Then the next one is the Km. And the big point with Km, so Vmax is that, it's Vmax. Okay. Km, the important thing here is this refers to substrate concentration. Okay. Substrate concentration. And it's the substrate concentration which gives you, let's do this one, 1/2 Vmax. 1/2 Vmax. It's the amount of substrate you have to add to reach half the maximal velocity. So let me just give you an example in the lab, okay? How do you do this and how does it make sense? Let's say we do the first experiment where we're adding substrate and we see, oh, okay, we finally reached Vmax. The enzyme cannot go any faster. And we'll just put a number to this. We'll say that Vmax is 100 units per minute. And then all you have to do is you go back here and just find out how much substrate did you have to get to. How much substrate did one have to add to reach 50 units per minute? And the amount of substrate you had to add is the Km. And the Km is the measure of the affinity of the enzyme for the substrate. That's what it does. 1 second.
Oh, you just, to reach Vmax instead of 1/2 Vmax.
Pardon me?
You didn't write the 1/2 Vmax.
Oh, I'm sorry. Thank you. That is the key point. Thank you. So what happens is when you have a very low Km, so look at the graph, okay? If you have a low Km, that means you're way down here. That means you have to add very, very little substrate to reach 1/2 Vmax. So a low Km means, low Km is a high affinity. High affinity between the enzyme and the substrate. Conversely, if you had to go way out here and add a lot of substrate before you even reached 1/2 Vmax, that's a very low affinity. So a high Km is a low affinity. People get that? Don't make it any more complicated than this picture right here. There's not anything more to it. All right, you should be familiar with, I'm not going to go into much detail here, but how enzymes get inhibited. There's 2 types of inhibitors, okay? There are competitive inhibitors and non-competitive inhibitors. Remember that competitive inhibitors bind to the active site of the enzyme. Competitive inhibitors bind to the active site. They compete with the substrate. That's why they're called competitive inhibitors. Non-competitive inhibitors do not bind to the active site. They bind at a site other than the active site. And they inhibit the enzyme by doing that. They bind at a site other than the active site. That is the classic example of an allosteric inhibitor. An allosteric inhibitor. A molecule which binds to a site other than the active site, but it inhibits the enzyme. That's an allosteric inhibitor. Okay. When you're looking at these graphs, there's a classic type of curve you see with allosteric effector. Anyone remember what that is? It's sigmoidal. Excellent. Very good. Okay, so the type of curve we were just looking at was just a hyperbolic graph. Whenever allosterism comes into play, you see a sigmoidal, an S-shaped curve. Okay? So remember from the last curve, it just went straight up and leveled off. Allosteric enzymes have a sigmoidal curve. Sigmoidal, S-shaped curve. And all this is showing you is where an allosteric activator changes the curve or an allosteric inhibitor changes the curve. Bottom line, what do you guys need to know? Sigmoidal curve, it's an allosteric effector, or it's an allosteric enzyme. And what do? Allosteric effectors bind at a site other than the active site and change the activity of the enzyme. They can either change it for better or for worse. They can activate the enzyme or inhibit the enzyme. So that was Biochem 2 in a nutshell. Okay, so if you see a sigmoidal curve, you should just automatically think of allosteric. Allosteric. Even if it's with respect to hemoglobin. Because then you think of the allosteric binding of oxygen. Okay, that's enzymes. Ready to move on? You guys still awake?
Yes.
Okay. Kind of. Let's look at the vitamins. So this is going to be the nutrition part right here. Yeah.
How much time do we need to spend on the different types of enzymes?
Okay, good question. The ones you want to know are the dehydrogenases, because those are oxidoreductases, so oxidation reductions. The transferases, because those are where phosphates are transferred. So that's either the use of ATP or the making of ATP. And the classic example there is going to be a kinase. Great question. Okay, so let me just write this down. Oxidoreductases. Okay, so you guys all understand that's an oxidation reduction, right? Okay, so this is just the class. The name of the enzyme, which always catalyzes one of these, is called a dehydrogenase. If you see the term dehydrogenase, you know it's going to catalyze an oxidation reduction. Because dehydrogenase just means a hydrogen is being taken off something. But if you take off a hydrogen, you have to put it on something. So that's, first you have your oxidation, then your reduction. So lactate dehydrogenase, classic example. Lactate dehydrogenase. Pyruvate dehydrogenase, classic example. All right, the next one are the transferases. The common name in enzymes, that's when you see the word kinase. And they add or remove phosphate. And what you want to think about is either ATP going to ADP plus PI, and sometimes that phosphate is put on another molecule, or the exact opposite example. ADP plus PI going to ATP. Okay, transferases transfer phosphates. ATP, you either use it or you make it. The third class is the ligases. And this is great for metabolism because when we look, there's only about 12 enzymes you need to know in all of Biochem 2. It doesn't seem that way when we go through it. But when you go back to the class, that's really what it is. And these are the 4 classes that they fall into. Okay? So the third class is the ligase. And that's just putting something together, putting 2 things together. And the type of enzyme name you're going to see is a carboxylase. Carboxylase. So what does that mean? It means it's adding carboxide or CO2. Okay, so ligases add CO2. Classic example. Let me give you a couple, okay? Pyruvate. I think this is the one given on your handout. Carboxylase. So we're getting off just a little bit here in the Biochem 2, but this is stuff which is good. Pyruvate carboxylase, and then acetyl-CoA carboxylase. These are both ligases because they add CO2. Let me give you the example of acetyl-CoA carboxylase. This is acetyl-CoA plus CO2 goes to malonyl-CoA. Does this ring a bell for anyone? This is the rate-limiting enzyme in fatty acid synthesis right here. Rate-limiting enzyme in fatty acid synthesis. Acetyl-CoA carboxylase. So what you guys need to do is put together, okay, carboxylase. That means it's a ligase. And then this particular one takes acetyl-CoA, which has 2 carbons. It adds a molecule of CO2 to form 3 carbons, which is malonyl-CoA. So anytime you see high levels of malonyl-CoA, that's telling the cell we're making fats. This is the product of the rate-limiting enzyme fat synthesis. Turns out pyruvate carboxylase, anyone remember what this enzyme does?
Pyruvate to oxaloacetate.
Beautiful. Pyruvate plus CO2, so the name is giving it away. Pyruvate carboxylase, acetyl-CoA carboxylase. Acetyl-CoA pyruvate carboxylase adding CO2. This goes to oxaloacetate. This is the major enzyme in gluconeogenesis. Gluconeogenesis right here. So now you've already got 2 pathways nailed down. Rate-limiting enzyme in gluconeogenesis, making of glucose. Rate-limiting enzyme in making fats. Okay, 2 pathways you've almost knocked off right there. And then okay, everyone seeing the connections here? The last class are the lyases. And they do the exact opposite of the ligases. A lyase is a classic example, is a decarboxylase. Decarboxylase. So they take off, they remove CO2. So those are the 4 you want to focus on. And when I go over the metabolic part for each of the big enzymes, will remind me, and we'll go back to which class they fall into. Okay. Okay, we're going to go right into vitamins, and this is the nutrition part. There are a lot of questions on vitamins. There are a lot of questions. Okay, they're not hard though. That's the good news. They're not hard. So here's, for those of you who have my sheet, it's right there for you. Here's what you need to think of for vitamins. You need to know what the vitamin is, what the coenzyme form is, coenzyme, what the reaction is involved in, and if there's a disease associated with it. And that's what you need to know for the vitamins. So B1 is thiamine. The coenzyme form is thiamine pyrophosphate. What's the reaction involved? Decarboxylation. Perfect. Decarboxylation. So this is involved in lyases, right? All the lyase enzymes are going to have thiamine pyrophosphate. And then the disease state, which you guys have heard a lot, is beriberi. No. So, if you look at this, you can see that there's a lot of questions which can come about vitamin B1. Okay, so here are the classic types of questions you see. What is the active form of thiamine? Or what is thiamine converted to in the body? That would be the coenzyme form, so pyrophosphate would be the answer. What is the disease state which results from a deficiency of thiamine? Beriberi. Enzymes, now here's what you may see. You may see an enzyme catalyzes the following reaction. And they'll write out a reaction, and what you need to pick up on is what's going on in the reaction. If you see a molecule of CO2 leaving, decarboxylation, thiamine or thiamine pyrophosphate are involved. Okay, so just make sure that you own this stuff. Yeah.
Do they draw out reactions?
Yes, they do. Yes, they do. So The ones they draw out are the classic ones in metabolism, and what are they going to be? The regulatory enzymes. Those are the ones they draw out. So those are the ones we'll review. Okay? And there's only like 12 of them. It's really not hard. Okay, so just with this thinking, B2, you guys, riboflavin, right? So is it worth it for me to go through this? I know you've had it in nutrition. Riboflavin, B2, the coenzymes of FMN and FAD. Now remember again, this can be FMNH, and this can be FADH2. So either oxidize, reduce, oxidize, reduce. So therefore they're involved in which type of reactions? Redox. Perfect. And no disease state. So just make sure. Now I do suggest you guys look back in your books and look at the structure of what riboflavin is. It's pretty unique, and you should just, before the test, just look at the structures. Don't memorize them, just, well, I guess you have to memorize them a little bit, but just get familiar with them, okay? B3, niacin. Again, precursor to NAD or NADH, and NADP and NADP. Redox. And what's the disease state, you guys know that?
Pellagra.
Yeah, pellagra. Pellagra. Is there a B4? Not that I know. B5. Okay, this is what? Pantothenic acid? Pantothenic acid. Now, what you need to remember here, whenever you see an acid, another way of saying that name is pantothenate. That just means the acid's dissociated. The reason I'm telling you is don't get confused if you see pantothenate and it doesn't say pantothenic acid. It's the same thing. Pyruvate, pyruvic acid, same thing. Lactate, lactic acid, same thing. All right, so don't. If you see the pantothenate and like, oh my God, I don't know if that's B. It is. It's B5. Okay? Pantothenic acid. This is unique in that it's not a coenzyme for an enzyme, but it's a structural component of something. And it's the structural component of coenzyme A. It's a structural component of coenzyme A. It's a part of coenzyme A structurally. So again, it would do you good just to briefly flip back in the book and look at the structure of coenzyme A, and right on that page they have it highlighted. They say this is pantothenic acid, that part of it. Coenzyme A, real big in metabolism, acetyl-CoA, malonyl-CoA. It just carries carbons around. It's acyl. Those are called acyl groups. Hydrocarbon chains are acyl groups, ACYL. Okay. Any questions on that? Just go right through them. B6. B6 is what? Pyridoxine. Pyridoxine. B6. Pyridoxine. Okay, what's the coenzyme? Pyridoxal phosphate. Pyridoxal phosphate. So another connection you should make there is you may see a question of what do the active forms of thiamine and pyridoxine, or what do the active forms of B1 and B6 have in common? They have a phosphate group. And they may say a high energy phosphate group is attached. That's just another way of saying it. Okay, this is key. You're going to see a number of questions on this, and based on what they do. These are the ones, aminotransferases. 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 And so what's the other name for them? Same thing, transaminases, aminotransferases, aka transaminases. I'm going to spend a little bit of time talking about these because they're important. Is there a disease state with B6? Look on your list.
No, there's no.
Okay. There's no disease state. Can't remember. All right. What is an aminotransferase? So I want to go through this reaction and then give you some examples because you'll see them in different forms. Aminotransferases take an amino acid, okay, so an AA amino acid, plus an alpha keto acid, so this is amino acid 1, alpha keto acid 1, and they go to amino acid 2 plus alpha keto acid 2. There's three, okay, so we're going to jump to metabolism because now I'm going to give you three classic enzymatic reactions catalyzed by transaminases, which do this. Okay, so you need to make some associations here. Okay. One of them takes glutamate, plus oxaloacetate, so here's amino acid 1, okay, glutamate, acidic amino acid. Oxaloacetate, I don't expect you guys to remember this, it happens to be an alpha keto acid. Glutamate, when it hands off, it's the amino group, that's why it's called an aminotransferase. It gets converted to alpha keto glutarate. So what you see is glutamate goes to alpha keto glutarate. Amino acid to alpha keto acid. Now you have the alpha keto acid here going to the second amino acid. The alpha keto acid always associated with oxaloacetate is aspartate. So what this is a conversion of an amino acid to an alpha keto acid, and then an alpha keto acid to an amino acid. So it's the same type of compounds on each side of the equation. They're switching. One amino acid goes to an alpha keto acid. One alpha keto acid goes to amino acid. Okay, this enzyme is called glutamate oxaloacetate transaminase. GOT. GOT. Glutamate oxaloacetate transaminase. GOT. When you do blood work, you look for the presence of this enzyme in the serum, and so a lot of times you'll see it called SGOT. That just means it's in the serum. Glutamate oxaloacetate transaminase. Remember, the enzyme names most often give you the substrate and then what happens to the substrate. And this just gives you both substrates. Okay. This is also called AST, just to make it confusing, because it's aspartate transaminase. So another name, this is equals, those equal the same thing. Serum stands for serum because when you look for these enzymes in the bloodstream, you're looking in the serum. Now, what's aspartate transaminase?
Aspartate transaminase is AST.
Okay. Now, why would I'm not doing this to torture you. Why is GOT important? Right. Exactly. That is the classic example of an enzyme which you look for myocardial infarction. If you have high levels of GOT in the bloodstream, it means your heart has become ischemic and those heart cells have lice, and this enzyme has come from inside the cell, and now it's in your bloodstream. And so that's why people, when you do blood work, will look for that enzyme when someone says, hey, I had chest pains. That's the enzyme you look for. Okay? There's one other classic one, and it's called GPT. GPT. Let's see if I can remember this. Glutamate. Okay? Glutamate is always associated with alpha-ketoglutarate on the other side. Okay, glutamate, when it loses its amino group, it always goes to alpha-ketoglutarate. When alpha-ketoglutarate accepts an amino group, it always goes to glutamate. Pyruvate is an alpha-keto acid. So it's glutamate plus pyruvate, and that's why it's called GPT, glutamate pyruvate transaminase. So here's the amino acid number 1. Here's alpha-keto acid number 1. Pyruvate goes to alanine. So glutamate again goes to alpha-ketoglutarate. Pyruvate goes to alanine. Amino acid going to alpha-keto acid. Alpha-keto acid going to amino acid. GPT. Hold 1 second. Just a few things. The reason this is so important is this is part of what's called the alanine shuttle. And this is a key point here. This will go back to the amino acids because alanine's an amino acid, right? Alanine and glutamine. Alanine and glutamine are the 2 amino acids involved in transporting nitrogen in the blood. That's a big thing that I forgot to mention in the first one. Alanine and glutamine transport nitrogen in the blood. Here is part of what's called the alanine shuttle, or the alanine cycle. And I suggest you just take a look at that in your book, okay? This is how excess nitrogen gets from the muscle to the liver via alanine. Why would you want to shuttle all your nitrogen to the liver? Because that's where the urea cycle is. So you can secrete it as urea. Perfect. Just knowing that, I tell you, that will carry you so far. So again, you've got to know some of these details, but put it into context. Put it into context. All right, and this enzyme is glutamate pyruvate transaminase. Absence or high levels of this in the blood, liver damage. Liver damage. No, of the enzyme. You're always looking for an enzyme. Okay, where did we start with this? B6 is a coenzyme for all of these reactions. B6 is a coenzyme for all of those reactions. Whew. Okay, cruising along. Biotin. Coenzyme form, biotin. Reaction, what's biotin always involved in? Carboxylation. Okay, so this is the coenzyme for the ligases. So this is a coenzyme for acetyl-CoA carboxylase and pyruvate carboxylase. Biotin is because, so flip back if you wrote that down in the page before when we talked about classes of enzymes, and I said acetyl-CoA carboxylase, rate-limiting enzyme, fatty acid synthesis. Pyruvate carboxylase, rate-limiting enzyme of gluconeogenesis. They are carboxylases. They carboxylate. The coenzyme there is biotin. Those are questions you'll see. What's the coenzyme for acetyl-CoA carboxylase? biotin, associated carboxylation. Okay? And they may just write out the reaction. But if you see a CO2 on the left side and another molecule on the right, it doesn't matter if you don't know what the hell the substrate or the product are. What the heck the substrate or the product? Erase that, would you? If you just see that CO2 disappear, that means it's being added to the substrate and going to the product, so it must be a carboxylation. Okay, even if you don't know what the substrate and product are. So your best guess that way is probably going to be right. Last but not least, vitamin C. The coenzyme, vitamin C. Okay, another name for vitamin C is ascorbic acid. Okay, this is really important in hydroxylation. Hydroxylation, so adding a hydroxyl group, an OH, to proline and lysine. And this is essential for collagen formation. This is essential for collagen formation. You must have what's called hydroxyproline and hydroxylysine to have proper collagen structure. And therefore, if you're deficient in vitamin C, you get scurvy because you don't make proper collagen. What we just went through are all the water-soluble vitamins. Did I miss any? I think that should do it. Okay, let's keep going here. Let's look at the fat-soluble vitamins. There's only 4 of them. These are fat-soluble. A, D, K, and E, these are the fat-soluble. High levels, okay, there's only 2 of these which you can, actually they can become toxic if you take too many of them. Does anyone know which 2 those are? A is one and D is the other. So that's a classic question. Which 2 fat-soluble vitamins become toxic if they accumulate too much? Okay, A and D. Vitamin A has a lot of different coenzymes. So there's retinoic acid. And retinol. We'll just stick with those 2. Let's go from retinol. It's involved in vision and rhodopsin. Okay. You need retinol to form with opsin to form rhodopsin. Okay, so the visual pigment in what?
Cones.
I think it's your rod. It tells you right there. And that's how I remembered it. Right now. Right, I forgot too. Yeah, it's your rod cells. So you get night blindness. You go night blind if you don't have it. And for the for time constraints, just take a look at retinoic acid. I'm not going to write it down. It's a proliferation of epithelial cells. But the main thing, vitamin A, retinol, vision, and if you don't get it, get night blindness. Okay? Vitamin D. What's vitamin? Coenzyme, vitamin D. What's the reaction? What's vitamin D important for? This is something you guys should know. Exactly. Calcium and phosphate. Levels. Okay? Main thing is calcium. Right? You guys should know that one for sure. Vitamin D is involved in calcium and phosphate metabolism. Focus in on the calcium. So what happens if you don't have a lot of vitamin D? You can get what? Osteomalacia, bone softening, what's the other one? Rickets, kids. Yeah, that's vitamin D deficiency. Vitamin K is vitamin K, and that's involved in blood clotting. Blood clotting. Blood clotting. We did forget a couple water-soluble ones. Get back to those in a minute. Okay, so this is involved in, if you guys gone through the blood clotting cascade, okay, thrombin, thrombosis, thrombin. Proper formation of thrombin is dependent upon vitamin K. And then vitamin E is an antioxidant. Vitamin E is an antioxidant. It prevents the oxidation of lipids, or the peroxidation of lipids. Okay? Yeah.
But the getting to all the conversion factors, more about hormone,
Oh, okay, great question. You should know the active form of vitamin D. Very good. 125 dihydroxycholecalciferol. That's the active form. Thank you. Vitamin D. Where is the active form made? These are good questions. You'll see these. The active form is made in the kidney. Where is it stored? Liver. And if you ever see storage questions and you don't know the answer, pick liver. Okay? Pretty much for any question, if liver is an answer, pick it. Because it does everything. And then also you can make a precursor to the active form in the skin. And you need sunlight to do that. So you can make the precursor in the skin, it can go to the liver, it goes to another precursor, and then it goes to the kidney where the active form is made. Very good question. Excellent question. Okay, what vitamin is involved in blood clotting, or what vitamin, question, a way of asking this is you may bleed out if you're deficient in which vitamin. So you just go, which one is involved in blood clotting? Okay. That's it for the fat solubles. So let me just do two more, and then I'll tie a couple together. The two water-soluble ones, which I forgot to mention, are folic acid and B12. And those are really important, and they go together.
Is that where you need to go through vitamin C as an antioxidant?
We'll get to that in just a second. Yeah. You can group them together, and there's an easy way to remember it. Okay. Folic acid, the coenzyme form is tetrahydrofolate. Tetrahydrofolate. This guy's important. Okay, this is a really important vitamin and coenzyme. So if you're going to spend some time on a vitamin, take a look at what this does. It's involved in the synthesis of purines. Okay, so that would be adenine and guanine. And also thymidine, which is a pyrimidine. It's also essential for some amino acids to be made. Okay, you can get, both of these will give you types of anemia. Okay, so what's the type with folic acid?
Megaloblastic.
Is it megaloblastic?
Yeah.
And then B12 is cobalamin. Cobalamin, and that results in pernicious anemia. So there are questions, these two will be bunch together. Which two vitamins are involved in keeping, your red blood cells together? It'll be better stated than that. Okay. Which two vitamins are essential for keeping your hematocrit up? Folic acid, B12. Really, take a look at this guy. Take a look at tetrahydrofolate, what that structure is, the enzyme which makes it, and just take a look through the book and what it does. That's an important one. Okay, that's a lot of material. What are the antioxidants? ACEs. That's what you need to remember. The antioxidants are ACEs. Vitamin A, vitamin C, vitamin E, and S, or selenium. Selenium. Okay? Selenium, vitamins are coenzymes. Metals are cofactors. So this is a cofactor. What's another classic cofactor you see in enzyme reactions? Magnesium. Yeah, zinc, magnesium. Perfect. Those are cofactors. Selenium is the cofactor for glutathione, the enzyme glutathione peroxidase. And let me show you what that enzyme does. So it takes H2O2, hydrogen peroxide, and converts it to two waters. Okay, the enzyme name is glutathione peroxidase. So selenium is a cofactor for that enzyme. It's absolutely required. And then, well, let's write that down. This is an antioxidant. It breaks down hydrogen peroxide, a free radical, high energy free radical. And my next question to you is, what is glutathione? Have you guys gone over that in any classes? You have? Okay. So what is glutathione? That's a question you'll see on the test.
Radical scavenger.
Okay, structurally, what is glutathione? Okay, very good. It's what's called a tripeptide. Tripeptide. So what does that tell you right there? How many amino acids? 3 amino acids linked together. And it's a thiol. So it has sulfur. So which amino acid? It's either methionine or cysteine. It's cysteine because of the SH group. Cysteine has an SH, methionine does not. It just has sulfur attached to a methyl group. I know I don't expect you to know that, I'm just telling you that. Take a look in your book, and all you got to do, I swear, you look in your book for 2 minutes and just, they have a picture of it, and you'll know it. Make sure, glutathione, and it's this guy right here, this SH. It can hand off the H over here to reduce it to form water. Okay, so glutathione's involved, works as an antioxidant, but it's not one of the ACEs. But that's where selenium comes into play. It involves glutathione and it involves glutathione peroxidase. Okay, 1:45. Wow. Don't think so. Listen, let's take a 5 to 10 minute break. And we'll pick up the pace next.
Okay. I can talk to you on break.
Yes. About 12:30. 12:30 will give us time to get through the metabolism. Okay, so I'm going to go. We'll take a half an hour to get through carbohydrates, fats, and DNA. Okay, so that's quick. So let's begin. On the carbohydrates, here's what you want to focus on, especially structure function. Just think monosaccharides, disaccharides, polysaccharides. Mono, di, and poly. The monosaccharides, there's only 5 that you need to know about, okay? Glucose, you know about. Galactose, mannose, fructose, and ribose. So let me tell you some similarities and some differences. The first 4, glucose, galactose, mannose, and fructose, they all have 6 carbons. Okay, so if you're looking for chemical names, it would be hexo something, because hexo is 6. Ribose has 5 carbons, so it's a pentose. So hexose, pentose, 6 and 5. So that's one thing you should know there. So what do you remember there? Way to remember it is just remember ribose is the only pentose. That's how it keeps it easy. Now, you need to know what the functional group is on these. They're all, so remember the exception. They're all aldehydes except for fructose. Fructose is a ketose. It has a ketone group. So everything else is an aldehyde. So fructose is referred to as a hexoketose. Hexoketose. Glucose is a hexoaldose, or an aldohexose. Ribose is a pentoaldose, or an aldopentose. Okay, so it's just the number of carbons and the functional group. They're all aldehydes except for fructose. So you should know, so when you study, make sure you know what a ketone is, make sure you know what an aldehyde is. It's very easy. Okay, disaccharides. Now we're just going to bring 2 sugars together. There's only 4 of them that you'll get questioned on. Okay? Every single one of them has glucose as one of the components. So that's very easy. The others, the names almost give it away. Well, at least for one of them. Sucrose, so that you should know this. It's just glucose and fructose. Glucose and fructose. That's what sucrose is. Disaccharide. Lactose, the name gives it away. It's glucose and galactose. Glucose and galactose. Maltose and isomaltose, both of those are just glucose and glucose. The difference is in how they're linked together. So the maltose has an alpha-1,4 linkage, and the isomaltose has an alpha-1,6 linkage. So go back and look at your book for that. Remember, the alpha-1,6 is the branch points. The branch points. Very quickly, I want to tell you what are the enzymes which break these down. Someone just give a guess on what would break down sucrose. What would sucrase, lactase, okay, maltase. So you'll see questions like that. All you have to know is this information, and you got it. The question will be, what are the 2 monosaccharides which are formed by the action of sucrase? You go, oh, that breaks down sucrose. Sucrose is glucose and fructose. It's that simple. Okay, you'll get. There's a lot of questions right here. Yeah, question? Yes, there is. Polysaccharides. That's plants and humans, that's all. This is right on your handout. There's only 4 of them again. Okay? Plants, you're going to have starch. Starch is 2 things, amylose and 2 amylopectin. All these polysaccharides, they're just glucose linked together. They're just glucose linked together. Great. We'll have some handouts, some pictures to look at this. 2 different types of starch. The third one in plants is cellulose. This is fiber. Dietary fiber. This has beta-1,4 bonds. We don't degrade glucose linked together in beta-1,4 bonds. has alpha 1,4. Amylopectin has alpha 1,4 plus alpha 1,6. Humans, we have one polysaccharide that we store, that's glycogen. That's a storage form of glucose in humans. Alpha 1,4 plus alpha 1,6. Okay, so what's the primary disaccharide form from the breakdown of glycogen? This is how you got to think. What's the primary disaccharide form from the breakdown of glycogen? Maltose, because alpha 1,4 bonds. The alpha 1,6 are just the branch points. Okay, there's many, many more alpha 1,4s. So that's the type of question you may see. Where does the digestion of sugars start? In the mouth. By which enzyme?
Salivary amylase.
Salivary amylase.
Or Tylen.
Tylen, right. The Tylen, however you want to say it. The digestion of carbohydrates is the only macromolecule which begins in the mouth. Remind me to talk about the digestion of proteins before we finish. Okay, some quick examples of these things I just talked about. Okay, here's the difference between glucose, fructose. Did I have mannose written up there? I did. Here's glucose. 1, 2, 3, 4, 5, 6. Hexose. This is an aldehyde group right here. C double bond O with an H. That's an aldehyde. Let's take a, okay, the aldehyde, the functional group, is always at carbon 1. Always at carbon 1. This is fructose over here. Fructose is a ketose sugar because it has a ketone group. And the ketone group is always at carbon 2. Everyone clear on that? Let's look at the relationship between glucose and mannose. Here's glucose, here's mannose. They're both 6 carbons. They both have aldehyde groups. The only difference is where the OH group is on carbon 2. It's just flipped. When you have sugars which are identical except for one carbon, they're epimers. Epimers. So that's another, they do ask those questions. Glucose and mannose are C2 epimers. That means the OH group is flipped at carbon number 2. Glucose and galactose are C4 epimers. Glucose and fructose are isomers. It's a more general class. Okay, and for the that should be described on your handout. Just so you can get this straight in your head, these are pictures of what's a amylopectin and glycogen. So these are storage forms. Amylopectins in plants. It's a starch. This is the storage form in humans. All these linear parts are glucose linked together in alpha 1,4 bonds. Anytime you see a branch, that's the alpha 1,6 linkage. Okay, so when you degrade this, when you start chewing up your sugars, the salivary amylase cleaves indiscriminately, just cuts alpha 1,4 bonds. So you're going to get much, much more maltose than you will isomaltose, because there's many more alpha 1,4 linkages than alpha 1,6. Okay? Is that clear? With amylose, amylose is just linear alpha 1,4. Cellulose is linear beta 1,4. These are the only two which are branched, amylopectin and glycogen. For whatever reason, it took like a week and a half to say that in Biochem 1. And it does, every time. Whatever reason. That's carbohydrates. That's the structure of carbohydrates. Let's talk about lipids. What is the primary form of lipid which we ingest? Triglycerides. What is a triglyceride?
Glycerol.
Glycerol backbone. 3 fatty acids esterified. Triacylglycerol or triglyceride. 3 fatty acids esterified to a glycerol backbone. If you remember that that is the primary fat we ingest, as well as that's how we store fat in the adipose tissue in the form of triglycerides. So therefore, so this will be just a little metabolism. When we break down fats we ingest, or we break down fats we have stored in our adipose, we break them down to a molecule of glycerol and 3 fatty acids. The fatty acids undergo beta oxidation to yield energy, ATP. The glycerol gets converted to glucose. And we'll talk a little bit more about that. Okay. What is the, what's a major lipid found in membranes? Phospholipids, great. So that is 2 fatty acids esterified to a glycerol backbone, but they have a phosphate head group. Okay, so a polar phosphate head group, and it's an alcohol head group. So 2 fatty acids esterified to glycerol, and then on the number 1 carbon is a phosphate head group. That will give you a phospholipid. A great supplement that people take is lecithin. What is lecithin? Phosphatidylcholine. Phosphatidylcholine, which is a phospholipid. So remember that. Lecithin equals phosphatidylcholine. You'll see that question. It's true. Every test I've reviewed has had that question. Every single one. Is phosphatidylcholine. It's just the common name for phosphatidylcholine, phospholipid. Okay. What are the essential fatty acids? Arachidonic, linolenic, and linoleic. Those are the 3 essential fatty acids. So the way I always ask this question on tests is which fatty acid can we make in the body? And I'll have those 3 listed and then one that I never talked about. Okay? And we can't make the essentials, so it has to be that other one. Okay? Just remember those 3 essential fatty acids. We have to take those in. Now, on the flip side of that coin is, what is the fatty acid? When we make fatty acids in the body, we always start out by making 1 fatty acid. It is the product of fatty acid synthesis. Which fatty acid is that? It's 16 carbons. No, listen. Those are essential. We can't make those. That's the whole point. Those are essential. We can't make them. So which one can we make? Palmitic acid. Palmitic acid is the end product of fatty acid synthesis in the body. That's probably not on your handout. Is that there, Leslie? No. No, okay. Palmitic acid is the end product of fatty acid synthesis in humans. It's 16 carbons. Fatty acid synthesis. Okay, what's the rate-limiting enzyme of fatty acid synthesis in humans? Acetyl-CoA carboxylase. Acetyl-CoA carboxylase. What's the product of that reaction? Malonyl-CoA. You see malonyl-CoA in the question, you think of fatty acid synthesis. What's the coenzyme? Biotin. Acetyl-CoA carboxylase. Biotin. The carboxylase tells you that's a carboxylation. You added CO2. Okay, Those are the connections you guys need to make. All right. What is a major, major important lipid in our body? Sterol. Give me the example of the sterol. Most common. You don't? Cholesterol. Okay. You need to know cholesterol. You need to know the structure of cholesterol. So if you need to know it, maybe I should show you. Cholesterol is the precursor to what? All steroid hormones. All steroid hormones. There's cholesterol. These 4 rings with this alcohol here, that's cholesterol. Okay, and it has this hydrocarbon chain, but these are the 4 guys you want to focus on. So I'll let you know, don't write it down, just look in your book, because those 4 rings are what you want to focus on. Cholesterol is the precursor to all of these guys. Testosterone, progesterone, okay, pregnenolone, aldosterone, cortisol. What do these guys all have? The 4 rings, okay? The 4 rings. So that's the key thing you want to focus on, steroid hormone. Quick thing on metabolism. How do steroid hormones interact inside the cell? How do they get in? Where are their receptors? No, okay, no, this is the exception. Okay? Steroid hormones, they're fat soluble, so they go right through the membrane, and their receptors are in the nucleus. Okay? The receptors for steroid hormones are in the nucleus. Very different than what you see with the hormones insulin and glucagon, which are what type of hormones are they? This is a classic question. They are which type of hormones? These are steroid hormones, polypeptide hormones. Insulin and glucagon are polypeptide hormones. Their receptors are in the cell membrane. Okay, so those are just good things to know. Got off a little bit. Okay, back to cholesterol. What's the rate-limiting enzyme for making cholesterol? HMG-CoA reductase. So anytime you see the word HMG-CoA, or that's not a word, but that abbreviation, think cholesterol. Just immediately. You see HMG-CoA, think cholesterol. HMG-CoA reductase is the rate-limiting enzyme of cholesterol. Here are the intermediates you need to know for cholesterol biosynthesis. Okay? Cholesterol biosynthesis begins with acetyl-CoA. Mevalonic acid, or mevalonate, is a big intermediate, and this is the product of HMG-CoA reductase. This is the product of HMG-CoA reductase. The enzyme makes this. And there are multiple steps going on here, but you don't need to concern yourself with that. I'm trying to think. You should definitely know squalene. An intermediate. SQU, yeah. Squalene and cholesterol. I have a feeling I'm leaving one out. Know that cholesterol synthesis begins from acetyl-CoA. 2 carbons, yeah.
So the only place that HMG acts is on acetyl-CoA for melanoma.
No, that's why, okay, here's the reaction catalyzed by HMG-CoA reductase. HMG-CoA reductase takes the substrate, so it gives it away. HMG-CoA is the substrate, and it uses NADPH to reduce it. That's why it's called HMG-CoA reductase. When you reduce HMG-CoA, it forms mevalonic acid. Huge point here about the coenzyme NADPH. Does everyone have this written down? No? Okay. HMG-CoA reductase catalyzes this reaction. I want to give you a very general but very important thing about NADPH. NADPH is used in anabolic reactions. NADPH is the reducing power used in anabolic reactions. What that means in general terms, NADPH is used to make things. Okay, so when I say NADPH is used in anabolic reactions, i.e. cholesterol biosynthesis, fatty acid biosynthesis, nucleotide biosynthesis. NADPH is the reducing power for those reactions. Fat cholesterol biosynthesis, fatty acid biosynthesis, nucleotide biosynthesis. So why don't, okay, so if you get this next point, it will help you out a lot. Why don't we use NADH and FADH2 to reduce in these anabolic reactions? What are they busy doing? They go to the electron transport chain to give us the energy to do these reactions. So NADPH does not go to the electron transport chain. We use this as reducing power. The NADH and FADH2 go to the electron transport chain to give us ATP, and we use that ATP so we can drive these reactions forward. You guys get that? That's a very big point. Okay, I'm going to move right along. The next class of lipids you guys need to know are the lipoproteins. The lipoproteins. That's the classic structure of a lipoprotein. Lipoproteins are used to transport lipids in the blood. They're used to transport lipids in the blood. There's 4 of them. Okay, and for those who have the handout, everything you need to know is right there. Okay. 4 of them are cholestyramine. Oh, Jesus, no. Chylomicrons, thank you. Chylomicrons, HDL, LDL, and VLDL. Okay, in a nutshell, chylomicrons are produced in the intestinal mucosal cells in response to dietary intake of fat. So you want to associate chylomicrons with dietary fat. When we take in fat, how does that fat get to the organs? Chylomicrons transport them there. Okay, so chylomicrons transport triglycerides to the organs. Okay, so if you go to McDonald's, you eat, fries and a Big Mac, all those saturated fatty acids, those triglycerides, will get transported to your tissues by chylomicrons. Yeah.
That's a little confusing because I thought that the triglycerides are broken down, then transported and built up again, once we get rid of those.
No. Triglycerides get broken down in your gut to fatty acids and glycerol. The fatty acids, well, it's close enough, okay? Fatty acids go into the cell, get remade as triglycerides, get packaged into chylomicrons, and then get taken to the tissue. The tissues then cleave off the fatty acids and use them. Yeah, so you're right there. Okay? Dietary. HDL. Okay, let's do this. Let's go right down to VLDL. chylomicrons carry triglycerides. Okay, they carry triglycerides. VLDL carry triglycerides. Okay, so that's the similarity. The difference is VLDL is made in the liver. It's like a third graders. So here's the difference, okay? When we take in dietary fat, chylomicrons transport it around. VLDL is made when we make fat. When we synthesize fatty acids ourselves, so when acetyl-CoA carboxylase is turned on, and we're making fatty acids, we take those fatty acids, the palmitic acid that we make, we incorporate that into triglycerides, and within the liver packages them as VLDL. The liver secretes them into the bloodstream, and then the VLDL will go to the adipose tissue to drop them off for storage, or it'll go to muscle to give the muscle some energy. Okay, so this happens in response to fatty acid synthesis within the body. Chylomicrons are dietary. HDL, high density lipoprotein, good cholesterol. Okay, 5 minutes. This brings cholesterol back to the liver. This is high density lipoprotein. Okay, brings cholesterol back to the liver. LDL takes cholesterol away from the liver. Okay, that's the next important thing. The density is the relative level of protein to lipid ratio. The higher the protein, the higher the density. And that's a question you'll see. They always ask that. Density refers to the protein-lipid ratio. Higher the protein, lower the lipid, higher the density. So HDL has much more protein and less lipid than LDL. Okay, lipids in a nutshell. Fats and lipids. Let's talk about nucleic acids, DNA and RNA. Very, very simple, okay? This is, DNA and RNA is a section where you do not have to devote much time to. Let's start with DNA. What's a nucleoside? It's a versus a nucleotide. Nucleoside is a sugar, a pentose sugar. Plus what? Nitrogenous base. So the pentose sugar, based on what we just talked about in monosaccharides, what must that be? 5 carbons, so what's the only one with 5 carbons? Ribose. Okay? So this is either ribose, okay, ribose for RNA, and deoxyribose for DNA. Okay, so the sugar is always ribose, and whether it has an oxygen at position 2 or not, it's either ribose or deoxyribose, RNA and DNA. What are the nitrogenous bases? Okay, purines or pyrimidines. So you have purines, or adenine, and guanine. And the pyrimidines equals cytosine, thymine, and uracil. are in DNA, and cytosine and uracil are in RNA. So whenever you see uracil, you think of RNA. Uracil replaces thymine in RNA. So a nucleoside contains a pentose sugar, it can be either one, and a nitrogenous base, and it can be one of these five. The only difference between a nucleoside and nucleotide is a nucleotide has a phosphate in it, plus a phosphate in it. Okay, so it has the pentose sugar, a nitrogenous base, plus a phosphate. So when you see the phosphates, think nucleotide. In DNA, it's double-stranded, right? And the nitrogenous base pairs do exactly that. They base pair. You always, in DNA, have adenosine, adenine binding with thymine, and guanine with cytosine. Always. Okay? Here are the purines. Here are the pyrimidines. They are bound by hydrogen bonds. I guarantee you will see a question about that. The question may read, how are the nitrogenous bases, how are two strands of DNA held together? Or which type of bond holds two strands of DNA together? The two strands, the double helix, okay, this is where the base pairs are. They're held together by these hydrogen bonds. The single strand is held together by a covalent phosphate ester bond. So covalent bonds, a phosphoester bond, hold the single strands together. When those two strands come together and intertwine, they're held together by the hydrogen bonds between the bases.
Could you repeat this single strand?
Yeah. The single band, it's a phosphoester bond, and that's a type of covalent bond. What else do I want to say? Oh, it's right at the end of your sheet. DNA to DNA is called replication. That occurs in the nucleus. DNA to RNA, transcription. That occurs in the nucleus. RNA to protein is translation. That occurs on the ribosomes, the rough endoplasmic nuclei. DNA to DNA is replication. DNA to RNA is transcription. mRNA to protein is translation. The first two occur in the nucleus, and the last one occurs on the ribosomes in the cytoplasm. Take a look at the structure of these guys. But you don't need to know much more than that. It's amazing how little and how basic it's kept on the boards. Okay? You know the purines, the pyrimidines, how they base pair, nucleosides, nucleotides, you'll do fine. The only thing you need to know is in RNA, okay, adenine binds with uracil. Okay, so the uracil replaces the thymine in RNA. So anytime you see this, uracil, or you see a U, think RNA. Someone had a question. Did you, Leah?
Yeah, I did. Kind of off the path here, but
It's okay.
just remind me, hydrogen bonds, I'm thinking of the tertiary structure of the proteins, and van der Waals and Larkin. What's the deal? They only fall under tertiary structure, those two. And are they important?
Okay, great. we just flew through carbohydrates, lipids, and nucleic acids. You guys ready to hit metabolism? Okay. See if I can get an overview page. Of course I can't. Oh, yes. Okay. We're going to dissect this baby in an hour, and you guys are going to know it cold. It's not hard, okay? That is my job, to convince you of that. I think I've just bitten off more than I can chew. Okay. So you guys know that I like looking at the big picture and then going into the details. What I want to do is talk about the hormones which regulate these pathways. The fours are 100 times easier than my tests. Okay? They're really, really straightforward. So the two hormones, well, there's 4 hormones that you need to concern yourself with, and 2 are major because they do the exact opposite. Okay, so they're going to be insulin and glucagon. So insulin, again, this is a polypeptide hormone. Insulin is secreted from the beta cells of the pancreas in response to increased blood glucose. Great. What do they do? What does insulin do? It increases glucose uptake. And here's where it stimulates anabolic pathways. So remember, there's 2 types of pathways, anabolic and catabolic. Anabolic means synthesis. Anabolic or synthetic. So what are the major biosynthetic pathways that we're going to talk about? There's only 2 of them. Really simple. Okay? One is glycogen synthesis. And what's the second? Fatty acid synthesis. Perfect. So just remember, in the presence of insulin, this only comes about when we have a lot of blood sugar. So when that blood sugar goes into the cells, we're going to do 3 things with it in this order. Okay? The first thing that happens in response to insulin is we stimulate glycolysis. Okay? Now I didn't write glycolysis down here because that's not an anabolic pathway. But the only reason we go through glycolysis, the Krebs cycle and the electron transport chain, is to get ATP. Okay, so we go through glycolysis, but the reason is to get ATP. And what do we use the ATP for? The anabolic reactions. Okay, so when glucose comes into the cell, the body, the cell or the organ would use it to get its ATP. And if there's excess glucose around, that ATP will be used to turn it into glycogen or turn it into fats. Okay, those are anabolic reactions. And the only thing which goes against that is glycolysis. But we need that just for energy. Okay. Conversely, the other enzyme is glucagon. This comes about from the alpha cells of the pancreas, the alpha cells, in response to decreased blood sugar. So what's the job of glucagon? Is to get those blood sugar levels back up and to get energy for the body. So glucagon stimulates catabolic pathways or breakdown pathways. What are the two that you need to focus on? Glycogen breakdown and fat breakdown. There's one little caveat here, in that fatty acid breakdown, triglyceride breakdown in the adipose tissue. Triglyceride breakdown in the adipose tissue is stimulated by epinephrine. It's not glucagon. So you want to associate glucagon with epinephrine. They really come about at the same time. They both stimulate catabolic pathways, and it just so happens that epinephrine is the one that stimulates fat breakdown.
Another tape?
Okay,
TV timeout?
sure.